University of Lagos, the Deputy Vice Chancellor, Academic and Research, the Deputy Vice Chancellor, Management Services, Deputy Vice Chancellor, Development Services, the Registrar and Secretary to the Government Council, the Provost College of Medicine, the Bursar, the Librarian, Dean Faculty of Dental Sciences, Deans of other faculties, Heads of Departments, all professors and other academic staff present, Your Royal Majesties and Highnesses, distinguished non-academic colleagues, dear students, past and present, men of the print and electronic media, all invited guests, distinguished ladies and gentlemen. It's my pleasure to welcome you all to my inaugural lecture titled Genome Editing, Surgical Editing, the surgeon-scientist narrative of Orofacial cleft research and care. According to Professor David Marks of the University of Bristol, an inaugural lecture is an unprecedented opportunity to showcase and summarize my research and career and to acknowledge the work of my close colleagues and mentors. It is also a great opportunity to thank my current research team and the wider group of academics that have collaborated with me over the years, and to celebrate our shared successes. Today, 18th of October 2017, what you're about to hear is all of the above. My research career began in the Department of Oral Maxillofacial Surgery in 1999 at Blue. Between 2003 and 2004, I was a clinical research fellow in the Department of Orofacial Surgery, University of Vienna, Austria. My undergraduate experience under the tutelage of Professor Orofacial, Professor Arotiba, as well as my experience at Lutz with Professor Gulewe, Professor Akiwande, University of Vienna under Professor Broda, University of Cologne, Professor Kubler, greatly influenced what I have become today, a surgeon of science. Those God has used to lift me up, these are the gentlemen and women. Professor Gulewe, Arotiba, Akiwande, and all these Europeans. During my 1 year sojourn in the University of Vienna Medical School, I was made to realize that it is possible to be a surgeon and a scientist simultaneously. There, I saw clinicians and surgeons combining clinical duties and responsibilities with basic science research. In addition, my PhD work at University of Cologne, Germany, opened my eyes to the possibility of being a surgeon and a scientist. In furtherance of my interest in genomics, I attended annual short course in medical and experimental mammalian genetics in Maine, USA in 2014. I was also in Murray Buccal Laboratory to learn and master tools, skills, and techniques in genetics and genomics, or Orofacial cleft. I was one of the few people that were nominated all over the world to attend the first ever international summit in human genetics and genomics. I enrolled for a professional certificate course in genetics and genomics, and I was awarded the certificate in 2017. The opportunity I had during my residency training, 1 year after in Vienna, and my PhD in Germany, and my training and research in genetics over the years made me what I am today, a surgeon scientist. Those who facilitated my journey in genomic research. As a maxillofacial surgeon, my clinical works and research revolve around the human mouth and face. And these are the range of diseases that I see. And these are the areas of my research expertise. But today, my narrative is all about my works in advancing cleft and palate research and care. Cleft and palate are the second most frequently occurring major congenital anomalies. Cleft and palate remains the most common congenital malformation in the head and neck region. And for cleft of the lid, you have opening in the upper lid between the mouth and the nose. If it is on one side here, if it is on height on both sides, you have this defect. For the cleft of the palate, is a gap in the roof of the mouth. And these are a range of phenotypes you see in cleft. How common is this defect? Every 2.5 minutes, somewhere in the world, a child is born with an orofacial cleft. There are about 170,000 children born with cleft of lip or palate every year. Not surprisingly, this birth anomaly is the single most commonly treated craniofacial malformation in a pediatric hospital setting. They represent a significant public health body in terms of immediate and long-term medical costs, as well as social impact on patients and their families. In the United States alone, the lifetime cost of treating orofacial cleft has been estimated to be approximately $100,000 US. Mr. Vice Chancellor, sir, these are some of the problems associated with cleft, as highlighted. These identified problems may be compounded by the limited availability of appropriate health facilities and resources and expertise, especially in our environment. Adeyemo et al. In 2016 reported that stigma or discrimination is commonly experienced by children of parents with orofacial cleft, and this can arise from family members, from friends, community members, healthcare workers, and even affected parents themselves, to the point that some of them actually kill their children. The quality of life and delivery of family-centered care can also be affected by poor access to specific information and lack of empathy on the part of health professionals. Mothers of baby with cleft and palate can go through emotional difficulties that make them perceive their role as being very stressful. This has implications on the overall emotional well-being. Formation of the lip and palate. This is the picture of a baby in the womb around 4 to 7 weeks of life. And these are the processes that must fuse if the lip is going to be formed. If they refuse to fuse on one side, you have this. And if they refuse on both sides, you have this. For the palate, you have around the 7th week in the woman's womb, in the womb, you have the tongue, then the two shells that form the roof are actually really vertical. But over time, because of growth, they raise up and you have the horizontal. And that's formed the barrier, that's the roof of the mouth, between the nose and the mouth. And in case they refuse to fuse, this is what you have. The formation of the lip and palate is a lot. The processes involve cell growth, migration, differentiation until you have the processes fused together. Cleft usually occur around the 5th week of pregnancy, before many women even know they are pregnant, especially in this environment where pregnancies are usually not planned. The cleft palate usually develops around the 9th week. Once the cleft has developed, it will remain throughout the pregnancy and will be part of baby's first smile. A cleft lip with or without cleft palate will change the appearance of baby's face. What are the epidemiology of orofacial cleft in Nigeria? Mr. Vice Chancellor, prior to our study in 2014, the regular that was a study that was published in 1982 used to be the most cited until our group published a new data in 2014 that shows that the prevalence is 0.5 in 1,000. However, we believe that this does not represent the true prevalence of cleft in Nigeria due to under-uncertainty. Many children are born outside the hospital in Nigeria, and evidence also suggests infanticide. Parents claim their children associated with orofacial cleft. Therefore, there is a need, Mr. Vice Chancellor, for a Nigerian population-based study. This is a big budget research endeavor that must be accomplished. We also examined the distribution of orofacial cleft in Africa using 34 countries. And these are the countries involved and the number of patients in each country. What do we find? In Africa, cleft lip constitutes 63% cleft of the lip, followed by 31% cleft of the palate. What are the causes of orofacial cleft? Orofacial cleft are usually classified either syndromic, that is those who have cleft with other bodily defects, or non-syndromic. The non-syndromic constitutes 70% and syndromic 30%. The syndromic cleft are caused by specific genetic aberrations, like this Van der Waals syndrome, and it has a characteristics bilateral lower lip lips with the cleft. On the other hand, the most common 70% is a genetically complex disorder caused by interaction of multiple genetic and environmental risk factors. And concordance, if you are talking about, you want to tease out the role of the environment as well as genes in any genotype or any disease, you look at twins. Concordance in identical use should be 100% if in non-zero, if only genetics accounted for the etiology. That means once one of the twins has it, the other must have it. However, what do we see in clinical practice? We notice that in only about 50% of the cases that you have the two of them present. Also, that is for identical twins. For non-identical twins, much lower, about 5%. What does this one tell us? This suggests a strong but not purely genetic etiology. In general, close relatives of cleft cases have a greatly increased risk of being affected. What about environmental factors? These are some of the causes in the environment. Smoking is the most consistent environment factor shown to increase the risk for cleft. Also, smoking among women increases the risk by 30%. Smoking 3 months before and 1 trimester are critical to the risk of having a child with cleft. Gene, genome, and orofacial cleft. Human body is composed of trillions of cells, and there are over 200 cell types in the body, and each cell contains 23 pairs of chromosomes. This is the chromosome. The cell-like structure of nucleic acid and protein found in the nucleus of most living cells. Each cell you have 23 pairs, and each chromosome contains many genes. And this is typical human karyotype with all the chromosomes. These are made up of DNA called deoxyribonucleic acid. And this DNA is packed into the chromosomes. In humans, these are elements of heredity. That means pass it from the father to the children, and from the parent to the children. These are elements of heredity that are transmitted from parent to offspring that contain biological information. In humans, these vary in size from a few hundred DNA bases to more than 2 million bases. The Human Genome Project, which was a very big project called Big Biology, which was completed 2002-2003, has estimated that humans have between 20,000 and 25,000 genes. Before this project, it used to be believed that we have about 2 million genes. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. And this is the DNA. Essentially consists of the base, the sugar, and the nucleotide. And the ATC, this A complements C, D complements C. A, T, C, D are the alphabet of the genome. And the other, the sequence of these bases determine the information for building and maintaining an organism, similar to the way in which letters of the alphabet appear in certain order to form words that we speak and understand. Human genome refers to the entire genetic constituent of human. And human genome has 6 billion nucleotides. More than 99% of those bases are the same in all of us. And each human is unique because these letters are arranged in a certain order, like this. It is the reason you are who you are, and why you are different from another person, the next person close to you. It is the reason the son resembles his dad, or mom, grandmom, or granddad. It is the reason why you have this different phenotype. It is also the reason why you have these different phenotypes. It is also the reason why some children are born like me, while others are born like me, all like this. It is the reason why some have sickle cell disease, bleeding disorder, cancer, hypertension, diabetes, and so on. While may contribute to the reason why some die young, while others live long. It is the reason some individuals react to some drugs, while others do not. It's also the reason some respond well to therapy, that's the treatment, while others do not. Genes and our genome have also been reported to influence our social behaviors. It's also the reason many of us must have observed that while a child resembles one of the parents early in life, but later takes after the other parent, many of us must have observed this. This is what we call genomic imprinting. A genome is an organism's complete set of DNA, including all of its genes and regulatory elements. DNA is the language the genome is written in. Genome is like an instruction manual that makes you who you are. Just like English language is written in A, B, C, D, ABC. and the Yoruba is written in RBD. The purpose of written or spoken language is to give instruction or communication. This well-regulated instruction is what makes one cell a skin cell, a kidney cell, another a brain cell. However, when you give instruction, you can have mistakes. Sometimes there's a mistake in the instruction manual. And this mistake, for example, can be point mutation, one of the 6 billion bases, or deletion, the chunk is deleted, or insertion, the chunk is inserted. How can we say that when you have a small change in 6 billion, how can it have serious effect? A single letter can make a big difference. A good example is what is common, many of us know about the sickle cell. Sickle cell is a result of a change in just one of the 6 billion bases. And you can see the profound effect due to that. For us to understand further, there is this common jokes on the internet to show you that a single letter can make a big difference. Let us, Mr. Vice Chancellor, let us consider this message. A man received a message from his neighbor. Sorry, sir, I've been using your wife day and night when you are not around at home. In fact, much more than you do. I confess this now because I'm feeling very guilty. Hope you will accept my sincere apologies. I will pay if you wish to charge service fee. And these 207 characters. Immediately after reading the message, the man shot his wife dead. A few minutes later, he received another message. Sorry, sir, spelling mistake. I meant Wi-Fi, not Wi-Fi. That these are some of the things implicated in the etiology of Orofacial. Mr. Vice Chancellor, sir. In 2006, Nigeria Orofacial Research Network was formed with the sole aim of identifying genes predictive of Orofacial clefts in Nigeria. This was later followed by African. Our central hypothesis was that genetic study for cleft and palate in African population can identify novel population-specific variants in candidate genes, because we know, as a matter of fact, that Africans are different. Our long-term goal is to identify meaningful genetic factors that predict cleft outcome in families. Why genomic study? Identifying those associated with cleft and understanding that the various contributing factors is an initial step in finding ways to decrease the prevalence of this disease. Ultimately, as research progresses, preventative measures can be taken, such as providing counseling for families possessing the disease, introducing modifiers like folic acid, and modifying nucleotide sequences using genetic engineering that I'm going to talk about later. Mr. Vice Chancellor, sir, our group have applied almost all approaches presently available in genetics and genomics. These include genotyping, target sequencing, replication study, GWAS, functional animal studies. Mr. Vice Chancellor, sir, our group was the first to publish studies on genetic and genomic etiology of cleft in Africa. We identified MSS1 gene. This is the gene located on the chromosome 4, short arm of the chromosome. In 2014, we also identified novel. When I say novel, novel means it has never been reported before. We screened DNA of 8 families with Van der Waals syndrome and 1 family with popliteal pterygium syndrome using Sanger sequencing. This gene has about 9 exons, but we only sequenced 4 exons. And we found novel mutation, nonsense mutation, splice site mutation, missense variant. In another study by Buccal Adeyemo et al. 2014, we discovered a rare functional variant in genome-wide association identified candidate genes for non-syndromic cleft. And this table shows that where you see 000 shows that these have never been discovered before. ADAP, PAC, and MAP gene. We subsequently investigated the association of risk variant in Europeans and Asians by GWAS, or by candidate gene analysis for association with cleft in cohorts from 3 sub-Saharan Africa. And we demonstrated nominal association with these genes. Part 7, the one in red shows the location, cytogenetic location. This is, for example, this is chromosome 1 short arm. This study was the first to demonstrate any of these association signals in any African population. We also sequenced GRHF3 gene for those with cleft palate only. And this gene is located on chromosome 1 short arm. And we also found 5 novel mutations. And these are 2 missense mutations, 1 splice site, 1 print and 1 nonsense mutation. These mutations were absent from 270 sequence controls and from all public exome and all exome databases, including 1,000 genome databases. However, 4 of the 5 mutations were present in unaffected models without the cleft, or the children have cleft, indicating that their penetrance is incomplete. This study provides evidence that as in Caucasian population, mutation in this gene contribute to the risk of non-syndromic cleft in Africa. We also observed 2 novel coding variants in this gene called GREM1, which is located on long arm of chromosome 15. These variants were not found in 194 African control mean those without cleft, and have never been reported in any public genetic variant database that includes 5,000 combined African and African Americans. In another study, in 2007, we sequenced all the 9 exons of IRF6 gene, and we also identified novel variants that have never been reported before. Genome-wide association study. It is an approach of massive screening of genomes of many individuals, the instruction manuals of humans, to look for genetic variants that are associated with the disease risk. It's a powerful tool in genetics and genomic research. To date, 7 GWAS studies for cleft and palate have been conducted, and 26 risk loci have been identified. Mr. Vice Chancellor, these studies have been limited to European and Asian population until we conducted our study. For the GWAS, our team was part of a consortium of 13 countries across 5 continents to report the result of GWAS of non-syndromic cleft palate only. And what did we find? We found a missense variant in this gene. We further examined this variant in zebrafish model and cell-based transactivation assay and conclude that this variant is an etiology variant for non-syndromic cleft palate. We also conducted a multi-ethnic GWAS on cleft and palate using 6,408 participants. Our GWAS revealed also novel association on chromosome 2 near this gene and on chromosome 19 near this gene. Mr. Vice Chancellor, sir, we successfully conducted the first ever GWAS study for orofacial cleft in Africa. And our findings for the cleft of the lip and palate. The significant traits were found on chromosome 3 and on chromosome 8. For cleft of the palate, the significant were found on chromosome 2 and very near significant on chromosome 7, 10, and 13. Our study has refined the genetic architecture of OFC in Africa, identify new loci, and demonstrate genetic heterogeneity for the clefts subphenotype. These findings will provide additional insight into preferential development and biology. Mr. Vice Chancellor, we also investigated the role of GJB2 gene, which is being implicated in non-syndromic hearing loss in some of our patients, and when identified damaging mutations in this gene, and that was found on the long arm of our chromosome 13. And the damaging mutation, the gene was mutated to VLIN. And using bioinformatics tools, we find that that mutation very digital and probably damaging. And this figure shows the chromatography, the change of cytosine to guanine. What about mutation in other human diseases? These are some of the mutations and the chromosomes associated with it. The one that's very common to all, that is very familiar to all of us is sickle cell anemia on chromosome 11, short arm. Today, there are 3 recognized treatments for genetic disease. Some well established, while others are still undergoing extensive clinical trials. And these are gene therapy, stem cell therapy, genome editing, or combination of the 3. Gene therapy involves the introduction of normal genes into cells in place of missing or defective genes in order to correct or prevent a pathologic process due to genetic disorder with the use of either a viral or non-viral vectors. And this shows the processes involving in transferring normal gene into the defective gene for the correction of the genetic defect. Examples of successful treatment include SCID, otherwise called bubble boy disease, immunoglobulopathies, inherited blindness, muscular dystrophy, etc. What about stem cell therapy? It's the use of stem cell to treat or prevent a disease or condition, including genetic diseases. These cells stem cells are cells that can turn to other types of cells. Bone marrow transplant for treatment of sickle cell disease, leukemia, lymphoma, and other diseases is the most widely used stem cell therapy. Stem cells are usually classified into 3, and the stem cell area is actually very controversial because of the fact that you need to manipulate the embryo. However, in 2006, Yamanaka and Takashi discovered that treating differentiated adult cells, including skin cells, with these 4 genes could differentiate cells back into an early pluripotent state. This finding has generated excitement based on the fact that almost any terminally differentiated cell can be coerced to convert back into stem cell without having to destroy an embryo. Mr. Vice Chancellor, genome editing is the precise modification of the nucleotide sequence, the 6 billion bases I was talking about. It's a type of genetic engineering in which DNA is inserted, deleted, or replaced in the genome of a living organism, including human, using engineered nucleases or molecular pair of scissors. This is molecular pair of scissors along any length in the 6 billion, and the nucleus cut, and you can delete or insert. What was once science fiction can now be considered science fact. The new tools of genome editing allow researchers to change the DNA of different organisms, including human, which can lead to changes in physical traits. Mr. Vice Chancellor, sir, these are the different genetic editing tools, but the one I want to talk about is called CRISPR-Cas9 for short. This is a new tool and has revolutionized the field of genome editing. It's a tool that scientists are currently using to edit or change the genome. It's an emerging gene editing technique with the potential to eliminate or disrupt HIV integrated genome or HIV infected cells from multiple HIV reservoirs, which will result in complete cure of HIV/AIDS. Presently, ethical issues and fears are being raised, both funded and unfunded. But we all remember the same happened at the inception of in vitro fertilization that was called test tube babies. But now, IVF and test tube babies are real. There are ongoing clinical trials using this editing tool for the treatment of sickle cell disease. The success of this trial will impact lives of many Nigerians with this disease. In fact, with CRISPR-Cas9 gene editing tool, we may be able to produce a Jurassic Park one day for those of us who have watched the film. Orofacial, cleft, and genome editing. Has cleft, lip, and palate, Mr. Vice Chancellor, sir, has cleft, lip, and palate been repaired using molecular pair of cells? The answer is no. Can cleft, lip, and palate be repaired using genome editing tool? Yes, potentially. Why is genome editing not applicable in orofacial cleft treatment for now? First reason, formation of lip and palate is a one-in-lifetime occurrence in a woman's womb. Once a child is born with cleft, it is impossible to use gene therapy, stem cell therapy, or genome editing to correct the defect, unlike sickle cell anemia, other immunoglobulopathies, muscular dystrophy, and so on and so forth. In addition, like I said, most of the ones that we see, 70% of them, are a combination of the environment and the gene. Gene editing may, however, have a potential usefulness in orofacial treatment in the future. Genome editing for the prevention of orofacial cleft may be possible for single gene Mendelian cleft, e.g. Van der Waals syndrome, which is an autosomal dominant trait. This is Van der Waals syndrome. It segregates in the family, passes from the parent to the child. These are autosomal dominant syndrome. Mutation in IRF6 gene. Some of the genes that we have also identified in Nigerians and in Africans. For instance, following pre-implantation diagnosis in IVF technique, mutation in a gene causing syndromic cleft can be identified, edited from mutant gene to the wild type. What is pre-implantation genetic diagnosis? Is a screening test used to determine if genetic or chromosomal disorders are present in embryo before you implant the genome. Once you find out that it is present, a CRISPR-Cas9 is used, you change from the mutant to the wild type, and that wild type is then implanted in the mother womb. Surgical editing. The purpose of genome editing is to use molecular pair of scissors to cut DNA at specific location, and thereafter remove, add, or replace DNA at specific site, with the ultimate aim of effect change in phenotype. Rather than molecular pair of scissors, we surgeons use surgical scissors and surgical blades to effect the change in phenotype or outlook appearance of this patient. Mr. Vice Chancellor, sir. Surgeons will use a combination of things to effect change from this baby, the cleft, and this is the result. Also for the palate, edit this using surgical scissors to effect this. Presently, surgical editing, surgical repair, is the gold standard treatment for cleft and palate. In the last 10 years, we have operated more than 600 cases at the Lagos University Teaching Hospital. For cleft ear repair, we employ either tenisiranda or milar rotation. Tenisiranda is my own preferred technique, and my boss technique, Professor Gulewe. Our preference is milar rotation advancement. And these are some of the cases that we have done. This is before surgery, and this is after surgery. This is before surgery, and this is after surgery. For cleft of the palate, we use several techniques, and this is before surgery, and this is after surgery. Repair of bilateral cleft. Traditionally, we repair bilateral cleft with Millard's technique, meaning that we have to bang the lateral component of this lateral component of the prolabium, in our experience, give unsightly scar, especially in the dark-skinned individuals, which are more prone to excessive scar formation. A modification of this technique, in our opinion, was necessary to achieve a more aesthetic result, especially in the group of patients. I developed a technique, a modification of Millard fork flap, to repair bilateral cleft of the lid where the lateral component of the prolabium are excised. With this technique, we avoided unsightly scar and excessive scar formation. And this is the baby after Professor Desanu has made the baby to speak. This is it. Marking, then the fork flap, elimination of the lateral element, suture the muscles, and this is the baby immediately after surgery. The result of the technique. This is before surgery, and this is the baby 5 days after surgery. This is another baby before surgery and about 3 months after surgery. This is another baby before surgery and after surgery. Another baby before surgery and after surgery. And this baby before surgery and after surgery. And this woman, nobody ever wanted to marry her. And the repair was done some 3 years back, and the patient came to our clinic, and this is the woman 1 month after the repair. The outcome of this technique was published in African Journal of Pediatric Surgery in 2014, and the article has been cited several times. With treatment of cleft and palate approved, we are able to have other research opportunity, clinical research, and we compare the quality of life of parents and caregivers of children with cleft and palate before and after surgery. And what do we find? We found that caring for children with orofacial cleft significantly reduces the quality of life of parents because it really can be very stressful. However, surgical editing or surgical repair significantly improves the quality of life of parents and caregivers of these children. We also look at an evidence-based paper published with Professor Desalo, Professor Akamu. We sought to determine blood transfusion requirements and factors influencing blood transfusion and cleft surgery. Usually a controversy between me and Professor Desalo. The patient will need blood, we believe that the patient doesn't need blood. Okay, Adeyemo, we need to provide an evidence-based study, and that would be, we found out that homologous blood donation prior to cleft surgery in patients with preoperative hematocrit of 30% or more is not necessary. For patients with preoperative hematocrit of less than 30, type and strain of donated blood should be adequate. In another study, where we look at assess the surgical outcome of cleft palate, Taiwan and Adeyemo, for cleft repair, we use plan score to assess the surgical outcome, and we found out that the complication rate of 14% that we saw is comparing to what is being reported in the literature. Like I said, two techniques that we use for cleft preliminary. So we compare these two techniques. Our study, however, did not find any significant difference in the surgical outcome of these two techniques. The expertise of the surgeon and preference, just like I prefer Miller and Professor Gulere I prefer Penicillanda, Professor Gulere prefer Miller. It's a strong factor in selection of the technique of repair in natural cleft repair. Mr. Vice Chancellor, Adeyemo also compare wound healing complication following the use of either absorbable or non-absorbable suture for skin closure and cleft. We found no statistically significant difference in the incidence of wound density and surgical site wound infection following the use of either resorbable or non-resorbable suture. However, more cases of tissue reactivity were recorded in the resorbable group. Mr. Vice Chancellor, Adeyemo described the technique and also the outcome of using Buccal fat pad as an adjunct flap in cleft lip repair. And the indications for those that have very wide cleft, for those with inadvertent tear, when you do surgery sometimes the mucosa may tear, and this is the harvesting of the Buccal fat pad, and this is why the Buccal fat pad is in situ. In all our cases, preoperative evaluation was satisfactory in all cases with healing of the flap and complete epithelialization of the BFP in 1 month. We therefore conclude that Buccal fat pad is a reliable adjunct flap in cleft palate repair. In 2004, I introduced the application of Buccal fat pad in orofacial reconstruction in Nigeria. This is a technique I learned when I was in here. And today, all papers written on Buccal fat pad in orofacial reconstruction, as of the last time I checked yesterday, in Nigeria are published by our research group. We also look at the role of genetics in third molar extraction. The third molar is a wisdom tooth. We genotype this particular slate with this G gradation on chromosome 17. And we found out that those with third molar impaction tend to have T and D rather than G at this locus. In this age, Mr. Vice Chancellor, in this age of evidence-based clinical practice and precision medicine, the decision to remove or not to remove a third molar prophylactically may be assessed in the nearest future on genotyping for these implicated slates. Mr. Vice Chancellor, like I said, I'm a maxillofacial surgeon. My expertise goes across many of the subspecialties in the world of maxillofacial surgery. And the topic of today is about cleft. However, these are my contributions in terms of the paper published in other subspecialties of orofacial oral maxillofacial surgery. And together, I've published over 165 papers in peer-reviewed journals, chapters in books, 2 books. In conclusion, sir, I've presented some of our research and clinical accomplishments. In clinical research, we have provided evidence from the lowest to highest level of evidence to guide clinical practice. In genetic research, we have applied almost all approaches presently available, from genotyping to target sequencing to replication study and to GWAS. We have also provided evidence for implicated genes in the etiology of orofacial cleft through animal study, functional animal study. We have contributed to knowledge and still contributed to knowledge on the genetics and etiology of orofacial cleft in Africa. Mr. Vice Chancellor, sir. We have identified genes responsible for orofacial cleft in Nigerian Africa. Mutation in some of these genes have been identified in other populations. A large number of them, however, of these mutations are, however, novel and unique to Africans and Nigerians. We have also identified novel mutations responsible for Van der Waals syndrome that may be amenable to genome editing in Africa. Based on the current knowledge and advances, it is possible to prevent Van der Waals syndrome with application of gene editing tools using in vitro fertilization technique as well as pre-implantation diagnosis. Currently, surgical editing, surgical repair, is still the best modality of treatment for orofacial cleft, whether lead or palate. And that we will continue to do to the best of our ability. Future direction in our research. I've mentioned, Mr. Vice Chancellor, I've mentioned several techniques that we have used and the tools that we have used. I'm happy to inform you that on the 17th of July, 2017, our group got an NIH grant to conduct all genome sequencing of Africans and Asian orofacial cleft case parent trials. This is huge. We are going to be looking into the 6 billion nucleotides. This will be the first whole genome sequence of orofacial cleft in Africa. In fact, it is indeed the first NIH-funded whole genome sequencing for any chronic disease or trait in Africa. We are also involved in orofacial cleft phenotype, correlating phenotype, the appearance with the genotype. And we have a lab in Louvre where we do 3D facial photography, leaf photography. This is me doing the leaf photography and taking the picture. And this is the ultrasound. And the result of this, you get to hear in the nearest future. Here by Dr. Olutayo James. Our recommendation. Dr. Olutayo James is my right-hand man in orofacial cleft research in Lagos University and College of Medicine, University of Lagos. Mr. Vice Chancellor, my recommendations. To improve the quality of life of family caregivers, individual-focused counseling sessions should be organized for caregivers soon after birth. Nigerian government should establish regional centers of excellence in orofacial cleft care to cater for training, research, and treatment of children with orofacial cleft. This is the standard all over the world, and our country should not be an exception. Previous studies on cleft have been hospital-based, like I said, Mr. Vice Chancellor. There is therefore a need for a population-based study. Genomic research and technology, as well as precision medicine, is changing the world. Therefore, I recommend the creation of Center of Excellence in Human Genomic Research in Nigeria. It is high time we introduce a physician scientist program into postgraduate residency training in Nigeria, in line with global best practice in postgraduate medical training. Mr. Vice Chancellor, I have heard the role of research grants in our accomplishments has been practiced in most parts of the world. I hereby propose a minimum of $10,000 or 5 million naira, whichever is higher, as a startup grant for 3 years for all newly employed researchers with PhD or fellowship in all universities in Nigeria. However, an approved fundable proposal is a must. This will help generate preliminary data to support external grants from NIH and other grant awarding agencies, as also it will make all of us very busy. The downstream effect will be huge and will create a new biomedical industry in Nigeria. The time to act is now. Let me start by giving thanks to Almighty Allah who has made this occasion possible. He is Al-Wahid, the one,